Hi everyone, thanks for stopping by my poster today. I'm Darcy Diesberg and I'm a fifth-year graduate student in the Vessel Lab at the University of Iowa. We've known for a long time that changes in beta amplitude in the brain relate to motor output, both during movement execution and during movement cancellation. But recent cross-species research has revealed that beta on the single trial level is really better represented as a transient burst-like change in amplitude and not as a change in sustained beta amplitude. And following up on this, some investigations of beta burst dynamics on the scalp have revealed that these changes that we see in canonical averaged beta during movement execution and movement cancellation really do seem to relate to single trial beta burst dynamics during movement execution and movement cancellation. In particular, we see that beta bursts decrease over sensorimotor cortex during movement execution, and that beta bursts increase over frontocentral cortex and sensorimotor cortex during movement cancellation. And in fact, these frontocentral bursts seem to relate closely in time to sensorimotor bursts, so that whenever movement cancellation occurs, frontocentral bursts closely precede a reinstantiation of beta bursting in sensorimotor cortex. So while these investigations have been really informative with regards to the beta burst dynamics on the scalp that relate to movement cancellation, by their nature, they can't tell us anything about the role that beta bursts in subcortical regions of the brain might play during movement cancellation, even though we know there are particular subcortical regions of the brain that are critical for movement cancellation, such as the subthalamic nucleus, or STN. We also don't know whether the progression of these beta bursts through subcortical regions of the brain align with existing network models of movement cancellation in humans, such as the model that proposes that the STN is responsible for net inhibiting the thalamus and thalamicortical loops during movement cancellation. So in order to address these gaps in our knowledge, the current study had two main aims. The first was to determine whether subcortical beta bursts relate to inhibitory control during movement cancellation, or action stopping. And the second aim was to assess whether the temporal progression of beta bursts through subcortical regions of the human brain align with existing network models of movement cancellation or action stopping. Now, as you can imagine, it's non-trivial to go about acquiring invasive recordings from the subcortex in human participants. However, we have the opportunity to do this during deep brain stimulation surgeries. So we recruited patient participants at the University of Iowa hospitals and clinics who were undergoing deep brain stimulator implantation in either the STN or the motor thalamus, specifically the ventral intermediate nucleus of the thalamus. And while these patients were awake in surgery, we had them perform an auditory version of a task called the stop signal task, which requires participants to execute movements and then occasionally attempt to cancel those movements. So participants receive a go signal on 100% of trials, which tells them to initiate a response, and then on 1/3 of trials, they receive a second signal that cues them to try and cancel that response. And while they performed this task, we recorded from the subcortical region of the brain where the DBS was being implanted, as well as from sensorimotor cortex using subgalial strip electrodes. So we recorded from the STN and the sensorimotor cortex in 9 patients, from the motor thalamus, or the VIM, and sensorimotor cortex in 11 patients. And in one particularly unusual surgical case, the patient was having VIM implants removed and STN implants placed. So in this patient, we were able to record from all 3 regions at once. So first, we looked at the beta burst dynamics across our recording regions during movement execution following the go signal. And what we found is that at all recording sites following the go signal, beta bursts decreased. So we see in the STN, in the thalamus, and in sensorimotor cortex, there is a decrease in beta burst rate, which is shown here on the y-axis of these plots, across time bins following the go signal, which is shown on the x-axis. Failed stop trials are shown in navy blue, successful stop trials in light blue, and correct go trials in yellow. For sensorimotor cortex, contralateral go trials are shown in orange and ipsilateral go trials in light green. We also found that the presence of beta bursts between the go signal and the response seemed to have an inhibitory effect on motor output, so that trials that contained beta bursts had slower reaction times on average than trials that did not contain a beta burst. And this is in line with previous findings that tell us that beta bursts might index inhibitory states in sensorimotor cortex. Now, one of our main aims of the study, of course, was to investigate the role that beta bursts play during movement cancellation. So in order to test this, we quantified the total number of beta bursts between the stop signal and participant stop signal reaction time, which is an estimation of how long the underlying stopping process took. We found that in both subcortical regions, in the STN and in the motor thalamus, successful stop trials contained more beta bursts on average than either other trial type, so for failed stops or matched goes. And importantly, this couldn't be accounted for by any pre-stop signal baseline burst rate differences across the trial types. So because we did see that subcortical beta bursts were elevated during successful stop trials, this indicates to us that beta bursts are indeed related to movement cancellation in these subcortical regions of the brain. Our next question was how these subcortical beta bursts might influence motor output and whether they might do so by influencing bursts in sensorimotor cortex. So in order to test this, we time-locked beta bursts in sensorimotor cortex to beta bursts that were happening in the subcortical regions of the brain during stopping. And what we found is that bursts in sensorimotor cortex demonstrated a peak in beta bursting in the 50 milliseconds following the bursts in the subthalamic nucleus during stopping. So this to us was an indication that the STN might influence motor output by upregulating bursts in the sensorimotor cortex during stopping. We also checked the temporal progression of subcortical bursts throughout subcortical nodes of our sample in order to see whether the progression of these bursts align with network models of stopping. Specifically, the idea that STN is recruited before the thalamus and net inhibits the thalamus. And we did find in our sample that STN bursts on average preceded bursts in the VIM of thalamus during stopping. So first we tested these relationships across our sample, understanding that these recordings came from different individuals. And we found that with respect to subject-wise SSRT, bursts on average occurred first in the STN during successful stopping compared to bursts that happened later in the VIM of thalamus. And while this timing comparison wasn't statistically significant when we looked at it across our entire sample, we also looked at this relationship within the one individual who had recordings from both the STN and the VIM. And in this single individual, we found again that STN bursts on average precede bursts in the thalamus during successful stopping, but not during failed stopping. And this was statistically significant in our single individual with recordings from both subcortical regions. And this is exciting to us because this provides evidence in favor of this network account that suggests that the STN net inhibits the thalamus during movement cancellation. So just to summarize what I've shown you so far, we demonstrated that beta bursts in the subcortex do relate to stopping, and in particular, STN bursts appear to affect motor output by upregulating bursts in sensorimotor cortex during stopping. We also found that the progression of beta bursts through the human subcortex provide evidence in favor of existing network models of action stopping. And in particular, this is exciting given the rarity of recordings that include multiple regions in the subcortex in humans. So thanks for listening, and if you have any questions about this work, please feel free to visit my poster. I'll be at my poster virtually on Monday, March 15th from 8:30 to 10:30 Eastern Time. That's poster session D. And also feel free to reach out to me via email or DM me on Twitter. You can also check out a preprint of this work, which we've now submitted for peer review. If you check out my poster, there's a scannable QR code that will take you right to this page on the bioRxiv. So thanks again for stopping by my poster, and thanks for listening.